going to be great. All right. Okay, I think Claudia, you are up first.
Hello everyone, it's a pleasure to be here today. My name is Claudia Biguetti and I'm from the Department of Bioengineering from the University of Texas at Dallas. And the goal of my research is to provide a new surface protein to mitigate dental implant failures. So there are so many causes of tooth loss, but mainly periodontal disease which affect about 20 to 50% of the global population. But we also can have, for example, trauma injury that leads to tooth loss and also tumor resection. And without teeth, people cannot eat properly, cannot even smile or interact. And we have a different approach to restore the oral health, even using prosthetic removable devices, dentures, or dental implants. And dental implants is the optimal treatment, the gold standard for replacement of teeth. So as you can see here in this example, we have a whole mouth being with a rehabilitation, and currently there is about 800,000 dental implants placed each year just in the United States. However, even this gold standard treatment have failures, and it is estimated that about 5 to 10% of commercially pure titanium implants will fail, and due to different reasons such as infection, factors related to the biomaterial, but also factors related to the host. And among the factors related to the host, we have aging and comorbidities like diabetes. So for improving dental treatments in this population, in my research, I try to understand what makes a dental implant successful, what makes the implant integrate with the bone. So when you put a biomaterial in our body, the biomaterial surface is interacted with our cells, but our cells do not recognize the biomaterial by itself, but the proteins that are binding to the biomaterial surface. And previous research has assumed this, which proteins are those? So bone proteins or blood plasma proteins, but we have about 3,000 proteins in our blood plasma, and doing this in vitro, it's not easy to guess which proteins are the main ones leading to integration. So what I did, I found a protein that is really important for healing, and I hypothesized that this protein would be important for integration of dental implants. And I did my in vivo approach and identified this protein. It is in higher levels in the protein layer surrounding the titanium implant when this is placed in the body. So then we hypothesized, okay, is this protein really important for osseointegration? What we did, we developed a model of oral integration in the mouse, and we put an implant in the mouse mouth, and then we inhibited that protein to see what's going to happen. And then what we observed was that here in the control, we have the implant and the bone surrounding the implant, and when we inhibited that protein, HMGB1, we observed that we got a failure, a fibrosis. So then we also did several assays, including gene expression, and we saw that inhibiting HMGB1, we mitigated the stem cell markers, also important for osseointegration. So now we decided then to deliver this protein in situations when we can have a failure. So for example, we are using the HMGB1 to mitigate failures in diabetes, and we have observed that in our models, even in vitro, in cell culture, or in diabetic mice, we are able to rescue the osseointegration when we deliver these animals HMGB1. So we hope this treatment, these finds will allow for better treatment in population suffering from healthy conditions. Thank you so much for your attention.
Wonderful. Thank you, Claudia. Do we have a question, please? And we will bring you the microphone. Yeah, excellent. Thank you.
Yeah, excellent. Do you know what part of the, I guess, what mechanism of healing that HMGB1 is impacting within impacting in the tissue?
Yes, so HMGB1, when it's outside of our cells, this is a protein that we have inside the cells, regulating gene expression. But when this is outside of the cell, it binds to a receptor called CXCR4, and this receptor is present in the stem cells. So HMGB1 actually is recruiting the stem cells to the site of healing. But there is also like pretty new findings showing that by this receptor, HMGB1 can also improve mineralization of bone. So actually there is several pathways that this molecule can target. And we are now investigating all these mechanisms. Thank you for your question.
Awesome. Thank you, Claudia. Well done.
Thank you.